14th and 15th, the annual science fair was held at Somerville High School. Students from the biology, physics, and chemistry classes participated in the event. The exhibit showed the purpose of the experiments, the materials and procedures used, the results and observations, and the conclusions. Judges scored the exhibits on scientific approach, original journal, thoroughness, ingenuity and creativity, and advancement to science. We talked to some of the students about their projects.
What is your project on?
I did the effect of dispersed yellow 3 on planaria activity and regeneration.
How did you decide on this project?
Well, I like to work with organisms because I find them interesting, and it has something to do with
alum and prima fluxes and polyelectrolyte.
Your sources were from outside and not from the high school here.
Yeah, it's mostly from libraries and the Northeastern Library, the Tufts University Library, and from the books that Mr. Lokudara had.
How long did you spend on this project?
With the research and everything, it will come to about 3 months, I think, because I started in December with researching. And at first I spoke to people from Battelle Memorial Institute in Columbus, Ohio, and I was looking for pollution, using enzymes to detoxify toxic waste.
More generations and use more amounts of X-ray radiation next year.
My project is to test the toxicity of selected antibiotics on E. coli bacteria. The effect of Coke and Pepsi on Ugrena.
My project is on the crown gall disease, which is a tumor disease that occurs in plants. My project is on the effects of E. coli on NutraSweet.
My project is concerned with monoclonal antibodies and anti-idiotypic antibodies, and what I wanted to determine was if there was a strain difference in various types of mice for the production of antidigoxin monoclonal antibodies.
Working on your project, did it hinder your other activities?
Sometimes, yeah, because when you're working on it, you're unable to go out as often. But I tried to go out, as often as I could. But when you're doing it, you get tied up, and it just takes a lot. It takes a long time to do it, especially putting it all together.
I did it during my February vacation, so I didn't have a vacation at all. I just went there at 8 o'clock in the morning and came back at 5 o'clock. So for the whole vacation.
It took up a lot of time to get, to type it and everything, so I'm not a very fast typer.
No comment.
Yeah, I mean, you have to give out a lot of your social life for doing your project, but it's worth it in the end, I think. No, I scheduled it so it would be in the right time.
Yes, it is very hard to do your regular homework and do a science fair project and to have a social life also. The only problem I did have was comparing with homework while I was doing the science project.
I did most of my work after school and during all of my vacations, so I guess it hindered it a little bit, but it was worth it. It usually is, because there's a lot to gain from doing science fair projects.
Welcome to the Somerville High School State Science Fair Winners Show. It's my pleasure to introduce to you and share with you this evening 4 young ladies who have participated at all levels of state science fair competition, and at this time I would like to introduce them to you. One of the things that you have just viewed has been a tape of the Somerville High School Science Fair. This fair, as you can see, is an integral part of our program. Many youngsters use that fair as a springboard to participate in the regional fair and then on to the state fair. Somerville High School this year was very proud, and the department was extremely excited about the fact that we sent 12 students to the state science fair. 9 of them were winners. I'd like to share tonight with you the experience of participating and listening to 4 of these winners. The first one is Pradeepa Saraparagasam. She is a junior. Pradeepa won first prize at our high school fair, got an honorable mention at the regional fair, which, by the way, was held at the Potter House Community School, and got a second place award at the state fair. Pradeepa is doing something unique, and I think at this time she would like to share it with you. Pradeepa?
I did the effects of naphthalene acetic acid and benzyl adenine on plant tissue culture. Naphthalene acetic acid and benzyl adenine are plant hormones, and they help growth of plants, and plant tissue culture is a type of plant cloning. What I did was I tried different concentrations of plant hormones on my petri dishes, which contains leaf sections of the African violet. And the African violet, I used the African violet because it grows quickly and shows cell growth very fast. I have some petri dishes that show cell growth and callus formation, and what I did was I tried different concentrations of these hormones, and I found the best concentration that showed results in less than a month, and that was 0.2 milligrams of naphthalene acetic acid and 2.0 milligrams of benzyl adenine. Now, plant tissue culture is a good method of propagation of plants because you can cultivate plants in a less period of time than that it takes for plants to grow when you plant them in soil with seeds. And this concentration helps it to show results in less than 1 month, and they produce superior plants. And I used the African violet, and my leaf section showed results in less than 1 month. But usually the concentrations the labs use takes at least 2.5 months to show results.
Thank you, Pradeepa. Pradeepa's project, I might add, is very unique, and I think you're going to see a great deal of interested college professors in this project. Pradeepa, as I said before, is only a junior, and we're looking for a great deal of rewarding experiences for her next year. Thank you, Pradeepa. At this time, I'd like to introduce our sophomore, and our sophomore is Elizabeth Bottinelli. And Elizabeth, this year, got an honorable mention at the high school fair, a 3rd place award at the regional fair, and quite impressive is a 2nd place award at the state fair. I might add that as a freshman last year at the Somerville High School Annex, Elizabeth got a 3rd place award in 1984, and this is a pretty prestigious accomplishment for her. I think at this time it's my pleasure to introduce Elizabeth. Would you please?
The topic of my science project is crown gall disease, and specifically the tumor-inducing principle of the disease. Crown gall disease is a tumor disease that occurs in plants, and it occurs through the use of Agrobacterium tumefaciens, which is a gram-negative soil bacterium. On this plant, you can see a tumor, and this tumor occurs when Agrobacterium is inoculated onto a wounded area of a plant after 48 hours, and the cells of the plant change and become tumorous, and the tumorous overgrowth occurs. The first step of my science project was to show that the tumors indeed do grow. The next step of my project was to show that the tumor could be grown autonomously, and I did this by placing the tumor on a Murashigin-Skoog basic culture medium, which contains basic nutrients, and it also contains auxins and cytokinins, which are plant hormones. These plant hormones help to continue the normal cell cycle of the plant. And I found that the tumor could grow on the Murashigin-Skoog culture medium. However, the tissue from which the tumor did grow on the plant could not grow on the Murashigin-Skoog culture medium. The last part of my experiment was to reverse the tumor-inducing principle. Now, the tumor-inducing principle is the cause of the disease. And this is a plasmid, and a certain section of it, known as tDNA, enters into the cells, and that causes the transformation process. I reversed this crown gall disease by heating the bacterium at 37 degrees Celsius for different amounts of times. And I found that when I inoculated this bacterium onto the plants after the incubation, they did not produce tumors. This project not only is good in agricultural fields, it also is important through cancer research. Anything that is discovered in plant tumor is also looked into in human cancer to see if there are any correlations.
Thank you, Elizabeth. I think by listening to Elizabeth just for a few minutes, you can see the magnitude of her project. Again, I can't stress too much the fact that she is only a sophomore, and hopefully she will take this project a little further next year and talk about Phase 3 and possibly Phase 4 as a senior. And of course, these are just, again, I can't stress enough, terrific springboards for young ladies like this to use for entrance to colleges. And in talking about entrances to colleges, I'd like to, if I can, excuse me, Elizabeth, introduce Amy Vellucci. Amy Vellucci is no new person to this show for the simple reason that she was on last year, and Amy Vellucci has some credentials that are just exceptional. First of all, this year I might say that her prize at the high school fair was an honorable mention. At the regional fair, a high honorable mention. But most important and extremely significant is the fact that she got a second place award at the state fair. And I might interject that last year as a junior, Amy, she received a first place award, of which she hopes to use this $1,000 Globe scholarship to enter the college of her choice. Amy Vellucci.
The title of my project is What Kinds of Bacteria Does Chlorine Eliminate in the MDC Water System? An investigation into the most chlorine-resistant bacteria, coliform. I became interested in my project since I have a swimming pool in which chlorine is used to eliminate bacteria, and in addition, my family has had some problems concerning drinking water in the past. Discoloration was one of them. The purpose of my project was to prove that coliform bacteria are present in our waters in great quantities, and not always eliminated by the chemical treatment process of chlorination. In a bacteriological analysis of water's composition, I used the membrane filter technique and the multi-tube filter technique, and in this way I tested to see how many coliform bacteria were present in 100 milliliters of each of my water samples. I tested samples from the 11 locations for which the MDC tests, and I also used tap water from my bathroom and my kitchen. And I found out that not at all times does the MDC test as effectively as it should, and that sometimes these coliform do exceed the EPA standards. I feel overall that chlorine is a reasonably effective chemical in eliminating bacteria in our waters. However, there could be some methods in the future that could be worked on that would be more effective.
Thank you, Amy. I think one of the things that I'd like to plug in from our point of view as high school teachers is the fact that in each case dealing with students on a science fair project, we like to make sure that there is some sort of a problem-solving type of experience that they're involved with. Many of these youngsters do establish the effects of X on Y. And this is exactly what we're teaching them to do, because when they get out into the big world, or they get into research later on in their life, this is exactly the format, the basic plan that they follow. Many times they set up a control component, an experimental component, and this is exactly what we're looking for them to do. Amy's project, if you view it very carefully, and maybe one of you people can zoom in on this, states exactly that. A problem is established. Types of questions have to be solved. Materials have to be acquired. One of the things that I'm really excited about when we deal with these youngsters is the fact that they all do these projects at home under supervision of their teacher, but many of them are farmed out to universities, the FDA, industry, and we must really thank them for this type of cooperative, collaborative effort. And the next person I would like to share this time with, excuse me, Amy, thank you again, is Heather Guerrero. And Heather has one of the experiences, to the best of my knowledge, of sharing her work with outsiders, working in industry, working in a laboratory, working in a hospital, and collecting all this business, and in fact putting everything together. We must apologize for not having Heather's boards here today. They happen to be at a marine science competition. One of the things that I'd like to share with you about Heather is the fact that she is a senior, and her track record is quite impressive also. And of course, this year she was a high school third place winner, a regional first place winner, and she'll be going shortly to the luncheon established by Cabot Corporation, her and her parents and her teacher. And again, finally, something extremely significant to us, a first place winner at this year's 1985 State Science Fair. I'd like to introduce Heather Guerrero.
I've been working on basically the same project for the past 4 years, and it's been dealing basically with monoclonal antibodies and improving their clinical efficiency. Monoclonal antibodies are just man-made antibodies produced outside of the human body, which are designed to attack and lyse specific antigens. And the reason why you'd want to even use them is because the human's inability to fight off all different types of foreign substances that enter their body. And the biggest one nowadays is cancer. The human just does not have the capability of producing antibodies specific enough to attack and lyse cancer. Whereas a monoclonal antibody, since it is produced outside of the human body, is designed to attack and lyse the specific substance and is injected back into the human body and will therefore go and kill it. In 10th grade, I basically established the fact that I could produce a monoclonal antibody and put it back into a therapeutic lysing system and have it lyse. In the next 2 years, I concentrated mostly on trying to improve that clinical efficiency. In 11th grade, I worked on microencapsulation, trying to find out if microencapsulating monoclonal antibodies had any effect on their ability to attack and lyse and their antibody concentration. And this year, I worked with anti-idiotypic antibodies, which just worked with different strains of mice, trying to figure out whether or not there was a strain difference in the production of monoclonal antibodies, which is very important because to a lab technician, they're working with time, and it's much better for them if they use the correct environment to produce their monoclonals since the experimentation is so new. And in the future, I guess when I go to school, I'd like to try to continue on with the research because it's very exciting and it's a new technology. And that's it.
Thank you, Heather. Many of you are probably wondering how come we have 4 young ladies here. Well, the reason why we have 4 young ladies here is that out of the 12 that we sent to the state fair this year, 10 of them happen to be young ladies. It's a wonderful sign of the times. It's certainly encouraging to faculty people that young ladies are getting involved in the sciences and the mathematics and the technology, and a number of them will be going on to engineering school or pre-med or business like that. I'd like to, if I can, share some few things with each one of these if I can, and maybe the rest of the girls can come in here, please. And one of the things I'd like to address Heather about is the fact that she's going to college next year. And where are you going to college, Heather?
Boston College.
Why did you select Boston College, by the way?
Well, I was very interested in Boston College because I like to pursue a career in international business. I was accepted to a few places out of state, and I was kind of wary about leaving, home to go to places like Ohio and things like that. But Boston College just really appealed to me. I don't know.
Does this mean you're really going to continue your project on a part-time basis?
Yes, I'd like to try to take some courses in biochemistry and immunology since that's what my project basically deals with.
Okay, thank you. Amy. Amy has something really great to tell us on the fact that she is going to a local university. But how did you get in there, Amy, and when did you decide to go there?
I had been wanting to go to Tufts University for about a year because of the excellent biology program that they have there. And I applied early decision this year and was accepted. I found out in January. And I'm going to major in biology there, and I feel that science projects really helped me to make this decision.
Okay, thank you. And I think one of our roles here is to make sure that everybody understands this. And science fair projects, as I've mentioned before, and I'd like to reiterate again, are an integral part of our program. And it represents for us a springboard, a terrific springboard to get into not only good colleges, but outstanding colleges. I wish we had the opportunity to share with you all the other students who did go to the state fair, but these 4 girls are terrific representations of exactly what we're trying to do at school. Excuse me for a second, Amy. Elizabeth, our sophomore, and who did extremely well as a freshman. What are your plans for the summer, Elizabeth?
This summer I plan to work on my project with a scientist through the Museum of Science mentorship program.
Could you explain what that mentorship program is?
Basically, they pair you up with a scientist who is of your choice, the project that you will do, and for 6 weeks in the summer you work with them on your project.
Excuse me, how did you hear about this mentor program? I'm not really that familiar with it. Could you give us some information on it?
I received a pamphlet in the mail and then called up the Museum of Science and set up an interview, and through the interview now got to meet another scientist who I might be working with this summer.
Would you like to identify this person?
Since I'm not positive I'll be working with him, I don't think so.
Okay, good, thank you. By the way, are you a member of the Science Museum?
My parents are, so.
Okay, thank you. Excuse me, Elizabeth. Okay, thank you. Pradeepa. Pradeepa is new to our country, and we're extremely proud of her. Would you like to share with us, Pradeepa, what your first gut reaction was to your science teacher about, listen, Pradeepa, you have to do a science project this year. What did you say to yourself?
Well, I did one in 9th grade last year, so it was new to me. But the projects were going to be like more advanced this in the 10th grade, so I thought I would explore more into science and see what I can do. And my science teachers helped me a lot with the science fair project, what I had to choose and how I had to get about doing it.
I see. Pradeepa's brother is also a participant at the state science fair. Did your brother help you at all?
No, he didn't. Well, I got help from Biotechnica, a lab in Cambridge, and a lady named Joanne Passiatori. She's a scientist. She helped me do my project.
Okay, very good. I think one of the things that I'd like to run over is the fact that these people do not perform in the classroom by accident. They perform in the classroom by accident is a total myth. They have outstanding teachers, and these teachers are constantly working with them. And in fact, I would go so far many times to say badger them about science fair projects. Could you give me an idea, Elizabeth, of what Mr. Slattery does with you concerning the total timetable of a science fair project?
Basically, he started telling us about them at around the middle of November, and I saw lots of signs in the building, Start Your Science Project Now. And when Mr. Slattery started talking to us about it, he gave us a little pamphlet on some of the science fair and what he wanted us to do. And he gave us some ideas, but basically you had to come up with everything yourself. And then we had a certain date in December when we had to show how much work we had done so far. And then at the end of February, our projects were due for him in our classroom, and we presented in our class. After presenting in our class, then we went on to our fairs.
Okay, good, thank you. I'd like to, if I could, just have Amy share with us some of her experiences about learning and coming across new friends at the State Science Fair.
I think the state science fairs are really good in that. They're not just good academically, but they're good socially, too. I met a couple of friends there that I still keep in touch with, and one of them, in fact, is in college now at the University of Hartford, and we still write. I think it's a good overall experience to go to the state fair, and I wouldn't have missed it for the world.
Amy, if you had to do it over again, what would you shoot for? Let's say if you were only a junior this year and you had to shoot for an award for next year, and you know the impact that your project has had on judges, and the level at which you compete, what would you go for? Would you go for another award like you have, or would you do something else?
I'd probably go for the National Youth Science Camp, which is a camp where all students go who are interested in science and meet together and socialize. I think it's something that would be an experience probably even greater than the Massachusetts State Science Fair. So instead of going probably for a scholarship, I'd go for something like that.
Okay, good, thank you. By the way, I might mention that Pradeepa's brother has in fact won that award and will be going to West Virginia. He will be going to West Virginia this summer, and he's one of 2 students in the entire state selected by Governor Dukakis to participate in this endeavor. Again, we're pretty excited about it. We were going to have Pradeepa's brother here, but he couldn't make it. Right, Pradeepa? Our mystery person on the left, you don't have to move, Heather. Our mystery person on the left, I'd like to just have Heather share with you some of her experiences about regional and state fairs, because she has been involved for a number of years, and I think she could give you a real good insight into what they mean to her.
Like as Amy said, science fairs have played an important part in my high school career, because they've given me the incentive to work in laboratories like the one I worked in this year, Mass General, and I've worked at Damon Biotech in the past. And it's just a good overall experience to set your mind to doing some work, doing it yourself, and over a long period of time, and just being able to present to judges and do wonderful television shows, and just express yourself, because you get more confidence, you meet more friends, and you just meet a lot of very intellectual people that you probably wouldn't have the everyday chance to meet, whatever.
Okay, thank you. Thank you. One of the things that we stress at school, and we recognize that it happens at school with reference to science fair projects, is the interdisciplinary business of it. I'd like to, if I may, thank you for allowing us to share this time with you. It has been a very enlightening experience for these students, and I know they won't ever forget it. If you have any questions at any time on any one of these young ladies' projects, please write me a note, care of Somerville High School. Joseph Pignatello, care of the Somerville High School Science Department. I think if you have any thoughts of your youngster or yourselves out there who are interested in science fairs, this is a good way for you to get some sort of an insight into exactly what we do and exactly what we hope to do in the future.